beginning level. What is cancer? Well, cancer is a cause where the actual cause lies within the body. Well, our body is made up of cells, and the cells which are normal cells present in our body, they can turn into cancerous cells by so many different ways. Every cell in our body has some programming done prior to it. What is that programming about? That programming is actually about how many number of divisions one cell will perform, what is the rate of that cell division, and after how many number of cell divisions that particular cell will die. This is the normal programming which is called apoptosis. So due to any reason, if that apoptotic property of a particular cell is lost, that cell will never die, and the rate of cell division increases by 3 to 4 folds. So this is how cancer develops in our body. Well, here I am mainly presenting one of my studies that we worked for an alternative research for the current scenario that is going on. Here, through this divine platform, I would like to present my alternative way that one of the studies that was carried out for this. So let me enlighten you with the current scenario going on. India, very rich culture. We are very proud of the prosperity, the culture, the devotion, the tradition that we all have. So if we talk about India, it's not just India, it's incredible India. When it comes to animal love, we worship animals as vehicles of God. We worship a mouse, a vehicle of Lord Ganesh. Simultaneously, an elephant is also worshipped at the same time. We worship a bull, a Nandi, as a vehicle of Lord Shiva. Simultaneously, there are a lot many birds that are worshipped, like we worship crow as vehicle of Shanidev. We worship a swan as a vehicle of goddess Saraswati. Likewise, when we talk about worshipping animals, how can we forget a holy cow? That is a symbol of so many different things. That is, a cow symbolizes a motherly love. A cow symbolizes, we can say, generosity, authenticity, and so many different things like wealth, strength, and so on. So all these things looking upon India, every coin has two sides. Let's not forget that animals are used in research purpose in so many different ways. When it comes to research, animals along with their products, they are utilized for the medical advancements, of course. Well, the range of animals which are used in research is varied, beginning from microscopic organisms like E. coli or viruses to macroscopic animals like some small size animals like houseflies, mouse, guinea pigs, to large organisms like pigs, even cows. Yes, you heard it right. Even the cows are used for the research purpose. But the main question that comes in our mind, are these animals actually required for the research? Well, giving it a scientific approach or a practical approach, taking into consideration the medical advancements, use of animals and their products, they have given us so many different utilities, taking care about, let's say, vaccine preparation, antibiotic preparation, and so on. The medical advancements that has increased the life expectancy by reducing the mortality rate, by vaccine preparations, and apart from that, so many different processes, the different animal products are used. For this particular thing, one of the findings from animals have made organ transplant possible today. So let's be more practical. apart from following the rules and guidelines, animals are surely required in research. But if we bring an animal at our home and if we bring an animal in our lab, well, the lifestyles of both the animals would be differing to a great extent. Yes, so when we talk about animals being utilized in a research, these questions can surely be flashed in our mind, that are we giving enough justice to this? Are we following some ethical issues and the emotions attached to it? So, here, we at our center were mainly or very keen to find an alternative, especially for an animal product, especially from cow. Here, we wanted, here the fluid that I am talking about, the body fluid that I am talking about is fetal bovine serum. Fetal bovine serum is used as a growth supplement for so many different research, so many different types of processes like cell culture, biopharmaceuticals, gene therapy and whatnot. So for this, let's throw some light on what exactly fetal bovine serum is. Fetal bovine serum, as I said, not only a growth supplement, but it is derived. How it is derived? It is definitely a growth supplement, and it is used as it is used for so many different processes. But how it is derived? It is derived from the fetal calf, especially when a cow is slaughtered in a slaughterhouse, especially the pregnant cows which are slaughtered. When we talk about this fetal bovine serum, how exactly it is derived? When a pregnant cow is slaughtered, it is injected directly, a puncture is created into the fetus's heart, and directly blood is aspired from there. Or it can be also aspired from major blood vessels to avoid certain contaminations and all. Once the blood is isolated, it has to surpass few of the processes that may include separation of the serum from the blood, heat inactivation and whatnot. Once all the certain processes are surpassed, that serum is fit to be used in the above mentioned processes. So let's not overlook the sad reality that how many cows are slaughtered for this. Basically, around 9 lakhs of cows are slaughtered in India on daily basis. The data of 2020 shows that about 3 crores 58 lakhs of animals are slaughtered. But this is for both the purposes, for the meat purposes as well as for the scientific needs. Our main focus was how can we improve the lifestyle of a cow, also focusing on their babies, especially where the research purpose is there. Can we find some alternative for this particular reason? Whenever we talk about something new, there are a lot many challenges waiting. The challenges that we have come across was that will the solution that we have come up with will solve this purpose, or that thing will mimic the result that the fetal bovine or fetal calf serum is giving. So here we present umbilical cord, a human umbilical cord, and the cord blood derived from that. So again some questions. Why this sample? Basically, this sample was primarily selected as the sample size was quite more. Second thing, the most important question that we came across is there any harm to the mother as well as the baby, especially when the process is done for isolation of blood, unlike in fetal bovine serum where the calf and the babies are killed. So the answer to this question is a big no. There is no harm to the mother and the baby. Why no? Because the answer lies within the process of isolation. The process of isolation of fetal calf or the fetal bovine serum begins when the cow is pregnant. This process begins after the baby is born. once the cord is separated from the mother and the baby, we can utilize this sample. But nowadays, the umbilical cord is preserved, we all know, for so many different reasons. So, can we still utilize this sample? And the answer is a yes, a big yes to us. Why? Because for preservation, only a small portion of the cord is necessary, and the major portion of that particular cord is discarded as a biological waste. So for this purpose, can we utilize this biological waste in some fruitful scientific measures? For that, we need to understand the structure of umbilical cord. The structure of umbilical cord, you can see it is a triangular structure. When we see the sectional view of that, it consists of two arteries and a vein, from which we can isolate a decent amount of blood. Those vessels, they are surrounded by a mucoid connective tissue that is called Wharton's jelly, which is a great reservoir of stem cells. Fine. The Wharton's jelly, basically it acts as an insulative covering on those vessels, and the name Wharton's jelly that was given on the scientist who discovered this in 17th century, James Wharton. So, what can be isolated from this? To get the best, we can take the blood from these vessels, and the Wharton's jelly is reservoir of stem cells. Now, what are stem cells? They are undifferentiated cells. Let me put some light on this undifferentiated word, what exactly it is. Yes, when a sperm and egg fuse together, it results in the formation of a zygote, which is a unicellular structure. When that zygotes divide to form a multicellular part, where all the cells are same, that is an embryo. But when that's in that structure, different organs are identified, it is called a fetus. How did this happen, that a structure consisting of same cells, and now we can see different types of tissues and organs? Do you agree that some cells would have changed their property and convert themselves into some other cell type, giving rise to these tissues? Yes, it is. So the stem cells have the ability to get converted, that is, differentiate into some other cell types. So stem cells can be isolated from Wharton's jelly, and the cord blood can be utilized for serum production. So for this, how exactly serum is made? But before moving to that, any idea how many babies are born? As per the data, around 67,385 babies are born daily. Well, this is a data of UNICEF. So, can we just consider this thing and put together with the serum that can be isolated from these samples? So, how is serum formed? Once the cord blood is collected into a vacuole or a test tube, definitely by maintaining the sterilized conditions or aseptic conditions, and it is allowed to clot. That blood clot, very simple thing, that when we are injured, the blood comes out, and after that, a dark brown net-like structure covers that wound. Who does that? Platelets. Yes, the thrombocytes of our blood, they cover the wound. When the blood is clot, the above supernatant is the serum. So, if per discard or that biological waste, we can isolate about 50 ml of the blood, out of which around 40% of that is serum. So, the purpose of this thing actually led us to fulfill this criteria. That is, the serum that was isolated from the cord blood was put together with the results with the fetal bovine serum that were nearly similar. So, what are the characteristics of cord blood? First characteristic, which is very important, they are nave cells. What are nave cells? Nave cells, basically, they are unexposed cells. Okay. Basically, umbilical cord is a connection that has been established between a mother and a baby while during pregnancy. Okay. That structure is attached to placenta. It is a tissue. It is a tissue that is embedded into the uterine wall. Through cord and placenta, all the necessary substances are transported from the mother's body to the fetus, and during the course of development, fetus generates lots of waste materials. Through the same part, they are given back to mother. How interesting it is that before even we are born, the dependency on our mother begins. So here, they are male cells. They are unexposed to any kind of tobacco smoke or any other nitrosamines or carcinogens. Second, they are actively dividing cells, which are exactly mimicking the results which are given by the fetal bovine serum. And lastly, the cells or the serum that has been isolated from a non-human source, may it be a cow, buffalo or a horse, can it cause some kind of antigenic stress over the human cells while developing a surrogate for cancer? Yes, it can cause. But the source is again of a human, so even that parameter has been removed. So here, what can we achieve out of this? Here we present 4,85,172 liters of serum per year if we can isolate all these births that are taking place on daily basis. How will this benefit to the government? Because Indian government, apart from producing serum, it spends a huge amount on importing it as well. So it can we can just have a cost cutting for the import part, importing also, and lastly, it can definitely save the lives of the babies, especially which are used for for the scientific needs. So this study mainly targets the researchers which are indulged into different types of researches in which one of the sample is fetal bovine serum. Apart from that, since the similar results have been shown by the cord blood derived serum, it can be used as an alternative or a better source for the fetal bovine serum. And the future plans will never stop because there is a long list of animals which are used in the field of research, and we would continue to find such similar ways which would directly or indirectly benefit either the animals or the researchers. Thank you for hearing me out. Namaste. Jai Hind.